to Medcast News, and today we're talking about organ rejection.
First off, there is no need to be afraid of a rejection of an organ, in this case the heart. Since 2005, 87.3% of the cases of transplantation have survived more than a year. This is an outstanding improvement since the 79.5% one-year survival in 1987.
Exactly. Today we have expert Maria Huertas on organ rejection.
Hi Maria, it is a real pleasure to have you today on Medcast.
No, the pleasure is mine.
So Maria, tell us a little bit about organ rejection.
So organ rejection is the response from the immune system attacking a new foreign organ, in this case the heart. So since the new organ has new antigens and different from yours, the immune system identifies it as bad and invasive, and this makes us attack it.
Yes, attack. So how does your immune system work in this case?
There's some cells that are called lymphocytes that are white blood cells and defend the body from disease and infections, and they also help the body against tumors. Two types of lymphocytes are B and T cells, and the 15% to 40% of the white blood cells are lymphocytes.
Okay, so what are T cells?
So T cells are so-called thymocytes, and they circulate through the thymus. And these are exposed to the antigens and they divide very rapidly. There's two types of T cells. There's the killer cells and the helper cells. The killer cells, also called the CD4 helper cells, assist the killer cells in performing their activity and help protect the body. This is also called cell-mediated immunity. And the killer cells, known as CTL, are essential in helping the B cells in destroy foreign substances, and 80% of the lymphocytes are T cells.
So what are B cells?
So B cells circulate the body through the blood, and they produce protein antibodies, and they attach to foreign proteins in the body antigens. They are found in the surface of microorganisms, and they help fight against bacteria. When antibodies attach to the antigens, this process leads to the death of the microorganism. And this is called humoral immunity. And B cells release antibodies into the fluid. B cells are activated and travel to lymph nodes and spleen. And 10% of the lymphocytes are B cells. The lymph nodes through this is that the immune systems include the B and T cells, and the lymphocytes circulate through the body to do their work. The dendritic cells infiltrate the transplant tissues by blood vessels, and the transport donor's antigens go to the recipient lymph nodes. When they're inside the lymph nodes, the antigens are represented to the receptors to helper T cells, and these helper T cells identify the antigen as a foreign. So there's two signals. The first signal is T cells that recognize the antigen of the cell because these cells display the surface peptide fragments derived by the pathogen's proteins. The foreign peptide are delivered to the cell surface by specialized hot cell glycoproteins that are then identified by a powerful effect on the immune system. And these antigens are called major histocompatibility complex, which are located on the surface of all your cells. And the peptide binding glycoproteins are known as MHC molecules. The antigens are bound to another molecule called MHC on the surface of all your cells. The MHC protein acts like hand holding onto the antigen so circulating immune cells can recognize it. Organ rejection happens when MHC tags don't match. And the T cells recognize the antigen only when it's bound to the MHC molecule. And then this cosmolutation, that it's the second signal. The binding of a T cell to an antigen is by itself not enough to activate a T cell. So in order to become activated by the T cell, must not only bind to the MHC peptide with its TCR, but also receive the second signal from the APC. This receipt of the second signal is called co-stimulation. When molecules of the APC designed B7 and their ligand to the T cell designated CD28, the binding of B7 to CD28 creates a second signal needed to activate the T cells. Then the immune system initiates the immune response by secreting cytokines including IL-2, And the IL-2 then binds to the cell surface receptors on killer T cells. This interaction sends activation signal to the JAK3 and the JAK1 enzymes located at the receptor base. The JAKs are non-receptor tyrosine kinases that activate signal transduction and activation encryption of 5 proteins. Travels to nucleus, and then it travels to the nucleus, delivers antigens to specific growth and activation signals, and then there's rapid cell proliferation that amplifies the immune response, and a new pack of killer T cells travels, and the antigen source proceed to attack and destroy, and this is how rejection happens.
When a new organ enters the system, the body mounts an immune response against the organ. T cells, which start the immune response, require two signals. One is the T cell binding to the antigen, peptide fragment, and the MHC. And the second one is the binding interactions between the T cells and the APC. Even though there are many pathways of amino acids selective against agents, CD28 is one of the best characterized receptors for immunosuppression. Immunosuppressive drugs block the binding between CD28 and the APC deactivating signal 2. Because signal 2 does not occur, the organ is not going to be attacked.
It's a pleasure to have you on set today.
Real pleasure to have you today on Medcast.